Hi everyone, and welcome to episode 25 of the Cancer Researcher podcast, brought to you by the European Association for Cancer Research. In this episode, we are directing our attention to the emerging field of cancer neuroscience. Increasing research efforts have been directed towards the study of peripheral neurons infiltrating the tumor and its microenvironment, as it becomes apparent at the moment that tumor innervation plays an important role in cancer progression, including for non-neural cancers. So far, the analysis of the interactions between peripheral neurons and cancer cells has been restricted to nerve endings. Today, I have here with me Dr. Andreas Trumpp, one of our board members, professor and head of Stem Cells and Cancer Division at the German Cancer Research Center, or DKFZ, in Heidelberg, Germany, and managing director of HI-STEM, the Heidelberg Institute for Stem Cell Technology and Experimental Medicine. I also have here with me today Dr. Vera Thiel, team leader at the DKFZ and HI-STEM. They will tell us more about Trace-n-Seq, a new technology they developed that enables the molecular characterization at single-cell resolution of neurons innervating any organ or tumor in preclinical models. We will explore the applications of this technology in pancreatic ductal adenocarcinoma, from deciphering neuron cancer microenvironment interactions to revealing their impact on tumor progression, and lastly, we will discuss the clinical implications of the discoveries made using this novel, cutting-edge technology. Let's dive into today's topic. Hi, Andreas.
Hi, Alexandra. Nice to meet you.
Hi, Vera.
Hi, happy to be here.
Thank you for accepting our invitation to discuss your work on the Cancer Researcher podcast. This episode was inspired by your recent paper, Characterization of Single Neurons Reprogrammed by Pancreatic Cancer, published earlier this year in Nature. Your labs have a long-standing interest in stem cells, so before discussing your paper, I would like to ask you what directed your attention to the study of tumor innervation.
Well, we realized that there is a lot of omics analysis of tumors. Actually, more than 10,000 cancers worldwide have been sequenced. And that will give us information about mutations that happen actually in the cancer cells. And if you do transcriptomic analysis, you also get information about the different cell types present in the tumor ecosystem. But what was lacking in all those data sets is the information of the neurons. And this is the reason why we got interested in neurons, because it was known since decades, based on work from surgeons, based on work from pathologists, that most of our cancers are also innervated by neurons, peripheral neurons if we talk about carcinomas. Now, what's in the tumor are the nerve endings, and the real cell bodies of these nerve endings are actually not in the organ where the tumor is, but they are in peripheral ganglia, such as the celiac ganglion or the dorsal root ganglia. So the nuclei containing DNA and RNA of the tumor innervating neurons are not part of the actual tumor, which is used to cut out in order to make omics analysis like next generation sequencing. And so we figured, well, it would be really interesting to develop methods to also characterize tumor innervating neurons at the molecular and single cell level. And to do that, Vera, here in the call, developed a method called Trace-n-Seq.
Exactly, yeah. This was, of course, the hard part, to identify these cells innervating these different tumors or pancreatic cancer, and to actually isolate the right cells, as the ganglia, of course, contain multiple neurons that can innervate different types of tissues and different types of organs at the same time. But we, of course, wanted to specifically isolate the ones that go either in the healthy organ or the tumor, respectively, which was then the hard part, and where we needed to introduce retrograde tracing into the method to really identify which of these cells actually projected into the pancreas or then in the tumor, respectively.
A major breakthrough that your paper brings is the development of the Tracer-n-Seq technology, which allows, for the first time, for the molecular characterization of tumor-infiltrating peripheral neurons in non-neuronal cancers. Can you please tell us a bit more about this novel technology and the way that it works?
Of course, yeah. So basically what we do is we inject a retrograde tracer directly into the organ of interest. In this case, we looked at the pancreas or the pancreatic cancers. And then this dye, basically, so we use the tracer fast blue, which is a dye that is then taken up by the axon terminals, which are present in the tumor microenvironment. And then this dye is retrogradely transported all the way into the corresponding ganglia, where it then specifically labels these cell bodies of interest which we want to harvest. And we develop these techniques of like harvesting the different ganglia subtypes, whether it is the sympathetic ones or the sensory ones that innervated the tumor. And we're able to dissect them, dissociate them, and then use flow cytometry analysis to basically sort these labeled cells out in comparison to the ones that are non-labeled and we're not interested in. Sounds now all very easy, of course, after the process is done. However, basically every single step was difficult based from the tracer injections, from which tracer to use, the right digestion protocols, because these ganglia are very delicate cells, and also then use a good flow cytometry to actually identify these cells, to then use them for downstream processing and to use them for multi-omic analysis.
And very importantly, we basically compare neurons which go into the healthy pancreas with neurons which project into the tumor. And then we actually compare what's the difference at the molecular single cell level.
So that's what I wanted to ask you next, because using these technologies, you said you looked both at the innervation of the healthy pancreatic tissue and several preclinical pancreatic ductal adenocarcinoma models. So what are the main differences that you observed between the innervation of the healthy and malignant pancreatic tissue?
We see actually a lot of different approaches or like different things in these pancreas versus pancreatic cancer innervating neurons. First of all, we see both when we just actually look at what we find in the tumor is that we find extensive sprouting of these neurons, which we could show are really only due to intense activation of sprouting, not due to infiltration by new neurons or any development of new neurons. Based on their transcriptomic level, we could also actually see that they start intense sprouting mechanisms. And the cancer is actually attracting also certain types of neurons rather than the others. So as we see great heterogeneity of neurons infiltrating the tumor in general, we also see a shift. Specific neuronal subtypes are rather needed than others. And then they change also when they actually innervate the tumor itself. So we see a lot of factors driving, again, axogenesis. We see a lot of neuropeptides change. We see actually transcription factors and metabolism genes changing. We also actually see a lot of immune regulators. So that was also a major part that we found, that these neurons also drive an immune suppressive phenotype in the tumor microenvironment. So we could see that the nerves interact with basically every single cell type in the tumor microenvironment. We could actually identify specific interaction partners and see that they are highly relevant, not just for the tumor cells themselves, but also for the other cells in the tumor microenvironment.
So my following few questions will address the clinical impact of your study. But before we move away from the ability of tumor infiltrating peripheral neurons to control the tumor microenvironment, as you alluded to earlier, I would like to ask you about the impact they have on tumor relapse, or whether there is an impact that innervation could have on tumor relapse. Although not many PDAC patients have resectable tumors, my understanding is that the primary potentially curative treatment for pancreatic ductal adenocarcinoma would still be surgery, and local reoccurrence of pancreatic ductal adenocarcinoma after initial resection is a significant concern in that case. Does the presence of tumor-infiltrating peripheral neurons impact on the ability of an excised tumor to relapse?
Yes, you're absolutely right. Sadly, for a lot of patients, surgery is not an option, but for the ones that is, this is, of course, very important to consider. And we indeed did analysis in which we could see that even if you actually resect a primary tumor in a mouse model, you could actually see these cancer-induced changes that happens in the nervous system are even maintained even months after the tumor was resected. So basically, they get into this specific cancer nerve state and at least partially maintain into this cancer nerve state. We can, of course, not know for sure if this changes stay your entire life. Therefore, more experiments would be necessary. But we do see that specifically after the resection, they maintain that state and can, of course, then still signal to the maintaining microenvironment in the specific state. We also did experiments where we injected secondary tumors, and we saw that nerves that were already primed into this cancer nerve state also induced a faster outgrowth of basically a secondary tumor, which mimics the relapse. And when we silenced these neurons, this effect was mitigated. So really showing that targeting the nervous system also beyond curative surgery might be very important in the clinics to keep patients also tumor-free for a longer time.
By translating this, as you already indicated, it's only about 20% of the pancreatic cancer patients which are eligible for surgery. But it becomes even more important that the surgeons take out the tumor entirely. This is often not completely possible. So it is, however, possible to then, after the surgery, sort of preventively block neuronal activation to inhibit outgrowth of a relapse.
There are also a few retrospective studies showing for a different variety of tumor types that patients who also take beta blockers, just as treatment for other diseases, often have a better outcome, less metastasis, and fewer counts of relapse, actually. So really showing also that this research is very important for tumor-free survival.
Thank you for explaining this so much better. We didn't get to discuss all the details in your paper, but one of the novelties that your paper shows is how these tumor-innervating neurons interact with cancer-associated fibroblasts. Now, cancer-associated fibroblasts can significantly impact the effectiveness of immune checkpoint inhibitors by creating an immunosuppressive tumor microenvironment. Do tumor-innervating neurons have an impact on PDAC response to immunotherapy?
This is still unclear. However, from data Vera generated, we have evidence that the tumor-innervating neurons indeed have an influence on the calves and actually promote their tumor suppressive function. Moreover, if we block the peripheral nerves, then these tumor suppressive calves change their fate and develop into pro-inflammatory calves, which lead then to the stream of incoming lymphocytes, potentially also T cells. And indeed, we did a small preclinical trial by combining chemotherapy like nab-paclitaxel with immune checkpoint inhibitors, and indeed found that immune checkpoint inhibitors, after blocking the peripheral nerves, would all of a sudden work also in pancreatic cancer. It's known that under normal circumstances, immune checkpoint inhibitors have no function in pancreatic cancer clinically. So these data, together with data where we actively also blocked in a more systemic way the peripheral nerves, we could show that this leads to the potential efficacy of immune checkpoint inhibitors. And we are currently planning a clinical trial addressing this in patients.
I think it's also very important to note that your paper also casts some light to further elucidate the mechanisms of action of some standard treatment regimens in the clinic. Moving on with the conversation, because you've mentioned wanting to move into clinical trials, I wanted to ask you, what are the next steps or future directions that you plan to pursue with this study?
Well, what Vera has shown is that by a systemic blockade of sympathetic as well as chemotherapy-mediated blockade of the sensory neurons, tumor growth is strongly inhibited. Moreover, as I just alluded, this may also lead to a better effectiveness of checkpoint inhibitors. So what we are currently planning is to block the celiac ganglion, and we will do that by a specific type of radiotherapy. Combine this with nab-paclitaxel, which we showed in the paper, would block sensory neurons. Combine this with gemcitabine, a classical chemotherapy, which is typically given in combination with nab-paclitaxel, and then add on top 2 arms plus minus checkpoint inhibitor. So this is a phase 1, phase 2 multicenter trial, which is headed in Heidelberg, but with other centers in Germany, where several comprehensive cancer centers will contribute to.
I'm looking forward to reading more about the progress that you're making with this study.
We are in the initial planning stage, so we expect that the trial may start to recruit patients by mid-26. So it will actually take probably another year until we can actually have that trial started.
Still sounds like a very promising avenue to pursue for pancreatic cancer patients.
In the meantime, we're of course also looking to extend our knowledge on cancer neuroscience, now going also a bit away from pancreatic cancer, looking at different cancer entities as well as their metastatic sites. If we find actually common changes between different tumor types, between their metastasis, to really see what impact these cells also have on different tumor entities. Because like Andreas said clearly in the beginning, these data sets are missing so far for basically every single tumor entity outside of the brain. So this is, of course, then very interesting also to hopefully target this in the future in a clinical trial after the preliminary experiments.
You basically use Trace-n-Seq to actually generate a pan-cancer atlas of cancer-innervating neurons to any tumor type in the trunk. So that includes breast cancer, pancreatic cancer, of course, but we already studied liver cancer, colon cancer, and many others.
It sounds like it might be good news for a larger range of cancer patients in the end.
We hope so. It certainly adds one important component of the tumor microenvironment, which was so far pretty much neglected or inaccessible at the molecular level.
But now with this new technology that you've developed, there's an opportunity for more researchers to come on board and help to further elucidate the role of the nervous system in cancer development.
That's absolutely right. And as you probably know, the field of cancer neuroscience is currently exploding, and worldwide, researchers as well as clinicians are very excited about these new options. And there are lots of activities to now integrate the peripheral nervous system also in the overall tumor ecosystems in patients.
Thank you very much to both of you for agreeing to meet with me today and tell me and our listeners more about your fascinating research.
It was a pleasure. Thanks very much, Alexandra and the EACR.
Yeah, thank you very much. It was a lot of fun.
We really hope that you enjoyed listening to this fantastic research story and that you learned something new from us today. If you have any follow-up questions regarding this conversation, please don't hesitate to contact the authors by email. Please subscribe to our podcast so you won't miss an episode. You can find The Cancer Researcher on your podcast platform of choice. Subscribe and tell your friends about it. And don't forget that you can also find us on Blue Sky and Instagram at Hello EACR, and you can search for us on Facebook and LinkedIn. We welcome feedback and suggestions for other topics or preferred guests through our feedback form, which you can find on our website at www.eacr.org/podcast. Until next time.